Thank you all for being here today. What is ESO? ESO stands for Intestinal Sulfide Overproduction. It's an increasing hydrogen sulfide producer such as the Sulfovibrio, Fusobacterium, and Proteus species, and some strains of Escherichia coli. And how the bacteria produce H2S? They produce it from cysteine or from sulfate reduction pathways. Previously, Dr. Villanueva Milan from our group had shown that hydrogen sulfide associates more with IBS diarrhea, while methane associates more with IBS constipation, and hydrogen sulfide associates more with IBS diarrhea. Moreover, she had shown that rats gavaged with the Sulfovibrio pyger and Fusobacterium varium showed an increasing H2S in stool compared to controls, and also an increase in stool wet weight. But what are the effects of H2S on cells? H2S can regulate mitochondrial function and dynamics. It also regulates gene expression, immune response, antioxidative response too, among other things. And we know that there is an association of H2S producers with disease, as you can see here. So far, no one has shown the profile of the small bowel transcriptomics in the context of ESO. So that's why we performed this study to identify the small bowel transcriptomics profile in ESO subjects from the RAIMAGIN study and determine if findings in ESO associate with findings in animals gavaged with H2S producers. A total of 85 subjects participated in the study. They were 18 to 85 years old, all sexes, and they were fasting. We collected a single and expiratory breath sample on the morning of their EGD or DBE. Then we assessed EMO by methane levels, and we also measured H2S. And we divided the ESO subjects into two groups. 1.5 to 1.9 ppm of H2S and higher or equal to ppm of H2S. Then we compared ESO versus non-ESO and ESO only versus non-ESO, non-SIBO, and non-PIBO. Moreover, we also collected small bowel biopsies to analyze transcriptomics, and we also collected small bowel aspirates for SIBO determination by culture. As you can see here, there was no change between the comparison groups in the demographics. And when we compared ESO 2.0 versus non-ESO, the transcriptomics profile showed more than 200 differentially expressed genes. Those genes were mostly associated with transcriptional and translational pathways, protein RNA pathways, and they were predicted to be upregulated because, as you can see here on the right side of this red line, they were associated with upregulated genes versus downregulated genes that are on the left side of this red line here. When we checked the pathways, we saw that response to oxidative stress was there as one of the top biological processes. And when we checked the genes, the differentially expressed genes, we had some genes of interest. The top upregulated gene was CYP1A1, and it's associated with an increase in reactive oxygen species production. At the same time, the top downregulated gene was humanin-like peptide, and humanin is known as an antioxidant. And we also saw an upregulation of several other genes that are also associated with antioxidation, except a downregulation of the glutamate cysteine antiporter, as we can see here. And when it's downregulated, it's associated with ferroptosis, that is a type of cell death. Then we thought, okay, maybe if we check H2S levels lower than 2, can we see some of those changes there already happening? And when we compared ESO 1.5 to 1.9 versus non-ESO, we saw more than 1,000 differentially expressed genes. Those genes were mostly associated with downregulation of immune response pathways, and we could see response to oxidative stress here already, and it was mostly predicted to be activated. Then we checked the cellular components, and it includes RNA, protein compartments, and also mitochondrial compartments, and we saw also secretory granules, extracellular space, and external side of plasma membrane there, and they were mostly associated with downregulated genes in immune response. When we checked the genes of interest, we saw again upregulation of several of the antioxidant genes and still downregulation of the glutamate cysteine antiporter. We saw downregulation of genes associated with gut motility and transit time, upregulation of genes associated with secreted gel-forming mucin, upregulation of TRPV1 that is associated with visceral hypersensitivity, dysregulation of acalpurines, and downregulation of prostaglandin receptors that are associated with inflammation and pain. Then we combined ISO 1.5 and ISO 2.0. We did the same thing. We found more than almost 900 differentially expressed genes. They were associated with RNA, protein pathways, mitochondrial pathways, and also downregulation of immune response. Cellular components included mitochondrial compartments, RNA compartments, and also secretory granules and extracellular space again. And the genes of interest included all the genes that I mentioned before. So we conclude here that these changes that we see here in ISO versus non-ISO, we suggest that they are really happening independent of the sample size because we put them all together and we still see those changes. Then we compared ISO only versus non-ISO, non-SIBO, non-IMO. We found more than 100 differentially expressed genes. Those genes were mostly associated with downregulation of immune response, as you can see here. Some interest genes included again CYP1A1 increased downregulation of the glutamate cysteine antiporter, and we saw downregulation of CTH, that is an enzyme that produces H2S endogenously in our cells. So we concluded first that when we focus on H2S levels in associated pathways, mitochondrial pathways are more associated with lower levels of H2S, while RNA protein pathways are associated with higher levels of H2S, and CYP1A1 increase is also more associated with increased levels of H2S. When we focus on conditions, when I compare ISO versus non-ISO, I see a little bit of antioxidant genes upregulated there, and this doesn't happen when I compare ISO only versus non-ISO, non-SIBO, non-IMO. Actually, we see downregulation of the glutamate cysteine antiporter that is also here, but on top of that, we see a downregulation of the endogenous H2S producing enzymes. Then we have two animal models here to show. I'll be very brief in this one, otherwise it will be long. But the first animal model, they were gavaged for 3 days with the sulfovibrio piger and Fusobacterial variant, and they were euthanized 2 weeks after the last gavage. These two bacteria are non-H2S producers, as I showed you previously. And we had another model of animal model where they were gavaged daily with E. coli BL21, E. coli K12, and Klebsiella pneumoniae, because we have previously shown that these bacteria are associated with small intestinal bacterial overgrowth. We gavaged them daily for 2 weeks, and we also gavaged them with Proteus mirabilis, that is another non-H2S producer, for 2 weeks. We collected small bowel from those rats for transcriptomics, and we analyzed the same way we did for the humans, and we collected stool to detect H2S levels. As I showed you previously, when they were gavaged with the sulfovibopyger and Fusobacterium varium, they had an increase in H2S in the stool compared to the control group. Moreover, they had an increase in stool wet weight. When we gavaged the rats with Proteus mirabilis, they also showed an increase in H2S versus the control group in the stool. And when we tested the production of H2S in Klebsiella and E. coli strains, E. coli K12 was the only one that produced H2S, as we can see here with these black dots. And the groups that received that were gavaged with K12 were the only ones to show an increase in stool water content. Then we analyzed the transcriptomics of all those rats, okay, and this will be a summary of the findings. So if you follow the colors, you will know what they dysregulated exactly. So for example, the sulfovibopyger and Fusobacterium varium, they were gavaged for 3 days only, okay, and euthanized 2 weeks after. The sulfovibopyger, most of the genes were associated with oxidative phosphorylation and mitochondrial respiration. Fusobacterium varium was the only clinical isolate from all these bacteria here that we took. We can see that there was a decrease in the cysteine, glutamate cysteine antiporter again. So we expect to see downregulation of antioxidative response because of that. There was a downregulation of the NRF2 gene, so we also expect to see a downregulation of antioxidative response. We saw dysregulation in transcriptional regulation, immune response, also in fluid homeostasis here. And we also predicted an increase in the adenylate cyclase activity, so we expect to see an increase in fluid going from the cell to the exterior of the cell. And if you remember the Fusobacterium gavaged rats, they showed an increase in stool water content in every single point that we measured. When we gavaged the rats with Proteus mirabilis, we saw an increase in net pH oxidase. We expect to see an increase in superoxide production. We saw also a decrease in oxidative phosphorylation and changes in RNA splicing. When we gavaged them with E. coli K12, we saw downregulation of several antioxidant genes. So we expect to see downregulation of antioxidative response and also dysregulation of mitochondrial dynamics. I forgot to mention that also Proteus mirabilis decreased 2 genes that degrade substance P. So if they are downregulated, we expect to see a upregulation of substance P there, accumulation, and also downregulation of immune response again. So we conclude here that ESO in humans and also H2S producers gavaged animals, they had changes in mitochondrial pathways in response to oxidative stress, in splicing and transcriptional pathways, immune response. They also showed changes in some genes associated with fluid homeostasis, nociception, and motility. When we gavaged the rats with E. coli BL21, doesn't produce H2S, we couldn't see any changes in mitochondrial pathways, we didn't see changes in oxidative stress, we didn't see changes in splicing pathways, we saw a few genes associated with immune response and fluid homeostasis. We didn't find anything related to nociception and motility. So we suggest that this is associated with H2S and strains matter. As future directions, we have planned to include additional comparison groups, spatial and single-cell transcriptomics, and also shotgun metagenomics. I'd like to thank the MASS program, all the participants in the study, all animals involved, and our collaborator, Dr. Ziliu from North Carolina State University. Thank you very much.